Hello, hi, a very happy good evening. So this is Krishnaveni and we've been talking about the story of life on earth, and that's biology. So welcome to my session. So today we are in lecture 3 talking about respiration in plants. So here today we are going to discuss electron transport chain. So we are done discussing about your glycolysis, your Krebs cycle. So today we are talking about your electron transport chain. So let's finish this off in style. So I will try to finish off electron transport chain because some students told me, ma'am, in this chapter glycolysis is easy, Tca is easy, but only electron transport chain is tough. So no worries, I will teach you step by step. Yes, Vairavan, a very happy good evening. Thank you so much for joining my session. So please do like, share and subscribe. So let your friends know so they will also get benefited because I teach fully in English. So you do not have a language barrier. If you want me to slow down, I can slow down, but please do share the link with your friends. Okay, so shall we go to respiration in plants? I will give you a recap of glycolysis, Tca, in between fermentation and your link reaction also. Okay, you just watched my first 2 lectures. Okay, so that's really nice. So thank you, Vairavan. Okay. So before we go into respiration in plants, so I have an announcement to make. So today we are having a mentee, so guess the chapter series. So it is episode 3 today. So please guess the chapter and play for my mentee. So the mentee quiz is at 6:30 p.m. So mentee will always be at 6:30. If there is a class, the class is at 6 p.m. Okay. Yes, Shimpi, a very happy good evening. I'm good, thank you. How are you? So please do like, share and subscribe, Shimpi. I hope Munna is good. Okay, so the mentee is at 6:30 p.m. It is guess the chapter and it is episode 3. So please do join me. So we are going to play for 20 questions and each question has a time limit of 20 seconds. So make sure you join me for the same. Yes. And this Saturday, 6 to 7, you are having a very different session. I have planned something very interesting for you. So hold on till Saturday. I will give you the announcement on Friday. Okay? Yes? So usually Saturday 7 to 8, we speak something outside biology, right? Something like motivation, something general. So this Saturday also, I have planned something very interesting for you. So hold on till Friday. So I will announce it on Friday. Okay, so Saturday we are going to have a very interesting session, evening 6 to 7, and it is not related to your biology chapters, so don't worry. Hi Keshav, yes, after a pretty long time, I hope you're doing fine. So where did you get lost in between? I hope you're doing good, and anyways, thank you so much for joining my lecture today. So come on guys, everyone, please do like, share and subscribe. So Shimpi says, fine ma'am, but I don't know why there is rain. It's good, no? You don't like rain? You don't like rain? Yes, it was drizzling when I was coming. So yes, I probably think after 10-15 minutes or by the time I go out after class, it's going to rain in Kota as well. So there's a lot of temperature change, right? It was really hot this morning. It was really hot last night. But now it's going to rain. But now when it rains, it brings in a lot of humidity, right? You sweat more. You can feel the heat more. So that is what happens. Okay, but at least enjoy the rain with a cup of coffee. So it's an online class, so if you're watching my lecture in a phone, so go sit by the balcony, look at the rain and look at my lecture, but don't miss the number of ATPs and glycolysis. Okay, yes, Anthony Abhi, a very happy good evening. So thank you so much for joining my session. So please do like, share and subscribe. There is a net problem. Okay, it's okay. So nature always takes the first hand, so everything else comes next. So if there is a net problem, so don't panic, just relax, close it, enjoy the rain with a cup of coffee, and after your net problem is resolved, so watch my lecture. Okay, and if you still have any doubts, get back to me. Yes, Anthony Abhi, I am very good, thank you. How are you? Okay, so today at 6:30, we are meeting again for Guess the Chapter series. It is season 1 and episode 3. So please go through the chapters. It is a chapter that you well know it. So 20 questions, 20 seconds each and 6:30. So before joining the session, so please do share the link with your friends. Okay, yes. So shall we move forward with respiration in plants? Shall we talk about glycolysis once again, Tca? I'm really depressed because they don't take any final decisions regarding board exam. In Kerala, they are going to conduct exam in September. Okay, so you're really frustrated and irritated because they're not telling you a deadline when exam is there, right? Yes, so every day you don't know you have to study or you don't have to study. So hold on, I have a beautiful idea. Okay, so if at all you don't drink tea, you don't drink coffee, oh my god, even I want to be like that, but I can't because I love tea and coffee. Especially I love coffee more than tea. But yes, anything that I get would suffice. That's not an issue. Okay, so Keshav, don't worry, don't be frustrated. So what can you do? Nothing is in your hands, right? So nothing is in my hands either. So it's going to happen, let it happen. So you cannot change what is going to happen, right? So yes, even if they say you're going to have your exam on so and so date, they will give you a time period of 1 month. So by now you would have read all the chapters time and again, and by now you would have been bored with all this, right? Yes. So they will give you a time gap of 1 month, and 1 month is absolutely enough for boards as well because you have a base. So don't worry, it's okay, you can do it. So don't worry, okay? So don't worry and concentrate on what you think you should learn. So read something fresh, so solve more questions, so your concepts become more strong. Okay, Keshav? So there is no point getting frustrated because everyone is helpless. We all thought Corona has ended, and that's when that was the hope that which we started year 2021. But see now what happened? It was twice the loss that we had in the last year. We never thought we will have twice the impact or the recap of 2020. But still we had. It is not there in anyone's control, right? So it's okay. So everyone has their own difficulties. Everyone has a problem at some stage. You might think, what is their problem? Why can't they do? Everyone fights their battle. Everyone has their own frustration. It's okay. Let it go. So as of now, you are safe, you're healthy, you are at home, you're getting 3 meals a day, you're getting exposed to a lot of other things. You are quite peaceful at the end of the day. So it's okay. There's no point getting frustrated and spoiling your health at this time. Okay, yes, I know second wave is most irritating and it has led to the lot of many lives. Yes, I agree, and my deepest and my heart goes out to all the grieving families out there, but we cannot help the situation, right? We can only stay faithful and hopeful. Even if they announce the date, they will give you a time period of 1 month. They will not say tomorrow is your exam. That's really not going to happen. They know the situation, okay? So don't worry, don't get frustrated. So read what you can, solve what you can. So improve well. So that is what you can do. Okay. So I drink milk. Okay. So the same my mom scolded me for drinking tea. Okay. So I am very crazy about milk, coffee, tea, etc. So yes, coffee, tea, I like coffee more than tea. But yes, if you give me tea, I won't say no. I will drink that also. Second wave, yes, I know. So Sanjeevani, hi. Yes, thank you so much for joining my lecture. So please do like, share and subscribe. I'm weak in 11th botany, how to overcome from that. Okay. So in botany, that means you're weak in morphology, anatomy and plant physiology, right? So these are your botany chapters, including your plant kingdom. So read your NCERT. So I have taught all the botany chapters. I have taught morphology more than once. Recently also I have taught anatomy also. Recently I have taught. Go to my lecture, see my lecture. In accordance, read your textbook, solve 25 questions in a day. If you still have any doubt, come back here and ask me, or there is a Telegram link beneath my description below my video. So please click on that. If you have a doubt, you can ask me instantly and I will get back to you. Okay, Sanjeevani? Yes, and thank you so much for faithfully coming and asking me your problem. I hope I answered your doubt. Okay? Yes. So guys, don't lose hope, don't lose your josh, don't lose your enthusiasm. So here we are studying everything in a slow pace. So imagine if NEET was not there, we wouldn't have had this casual time where we would have relaxed every topic in neat detail. We would have been in a hurry. So I would have been in a hurry to teach you a lot of things. You would have been in a hurry to read. But yes, by God's grace, whatever happens, happens for good. Okay? So shall we move forward with respiration? Here we go. So this is lecture 3 of respiration in plants. So moving forward, so what are the topics that we were discussing? So do plants really breathe? So do they have respiratory organs? So we answered all these questions. So then we went on to talk about your glycolysis. So this was followed by fermentation and your aerobic respiration, that is your Tca cycle. So today we'll talk about your electron transport chain. And finally, the substrate oxidation. So let's talk about ETS. So if time permits, we'll go to the next part. Otherwise, we will revise glycolysis, fermentation, aerobic respiration completely. Okay? Yes? Fine. So we will start with glycolysis. Nothing before that, nothing after that, because that is where the important points of this chapter starts. So tell me, what do you know about glycolysis? So glycolysis happens in all living organisms, irrespective of whether they are aerobic or anaerobic. So all living organisms undergo glycolysis process. So before that, what is respiration? The combustion of your glucose molecules to produce energy, right? The complete combustion, the complete breakdown of glucose molecules to produce energy is your respiration. So why do we need energy? To perform our day-to-day activities. Hi Pooja, yes, a very happy good evening. So thank you so much for joining my lecture. So please do like, share and subscribe. So Pooja, did you see my physiology videos? Did it help you? Do you have any doubts? Yes, Vairavan, you're absolutely right. Okay, so glycolysis is the first step in the process of respiration, which happens in all living organisms. Right, so where does glycolysis takes place? So glycolysis takes place in your cytoplasm. And one more thing, it is also known as your EMP pathway because it was discovered or deciphered by 3 scientists, Emden, Mayer, Parenhoff. So that is why it is known as your EMP pathway. So glycolysis is a 10-step reaction. So what happens? The initial substrate is glucose. So glucose is converted into pyruvate, into 2 molecules of pyruvate, right? So this is what happens in glycolysis. So what is more important is your substrate level reactions. So what is your substrate level reaction here? So glucose to glucose 6-phosphate. So what is the enzyme here? Hexokinase, right? And here you also need a cofactor Mg2+. So here your ATP is being utilized and ADP is getting produced. So this is one substrate level phosphorylation. So after this, what happens? So your glucose 6-phosphate gets converted into fructose 6-phosphate. So with the help of the enzyme isomerase, right? Now your fructose 6-phosphate gets converted into fructose 1,6-bisphosphate. That is, one more phosphate group is getting added. So this is catalyzed by the enzyme phosphofructokinase. So once again, here you need your Mg2+ cofactor, and here your ATP is utilized and to produce your ADP. So this is your substrate level reaction because you're utilizing your ATP, right? So remember these two. So what is the net gain of ATP? So how many ATP are used in glycolysis? 2 ATP molecules. So how many ATP are generated or produced in your glycolysis? 4. Because you get 2 molecules after one step, everything is 2 molecules, so you get 4. So the net ATP is 2. Right? And how many NADH here we have? So we have 2 NADH. So 1 NADH is equal to 3 ATP molecules. So the net ATP requirement therefore is 2 plus 6, so 8 ATP in case of your glycolysis. Done? Yes, you're absolutely right, Vairavan. So thank you so much for remembering everything in detail. So this is 8 ATP in your glycolysis. Phosphorylation means addition of phosphate group. Phosphorylation, so adding phosphate group to the substrate is your phosphorylation. Okay, Kesha? Yes. So this is your glycolysis process. So after glycolysis, at the end of glycolysis, you get 2 pyruvic or pyruvate molecules. Now this pyruvate can have 3 fates, right? So pyruvate can be converted into alcohol plus carbon dioxide. So this is your alcoholic fermentation. Yes, and this is in case of your yeast. So the examples are also important, okay? So then it can get converted into lactic acid plus carbon dioxide. So this is your lactic acid fermentation. So this can happen in case of some bacteria as well as in your muscle tissues. Done? Yes. So moving further, the third fate of your pyruvate is your aerobic respiration. So aerobic respiration happens in case of only aerobes, the one that requires oxygen to breathe, right? So this happens in case of aerobes. So here it is broken down into carbon dioxide plus water. So this happens in case of your aerobes. So here I have discussed glycolysis, I have discussed fermentation as well. So two things I have discussed. The next thing is your link reaction. So let's go to the link reaction. So this is the end product. So now we are going to talk about your aerobic respiration. So before we go to aerobic respiration, okay, so we are going to talk about the link reaction. So this is a link between glycolysis and your Krebs cycle. So this is a link between glycolysis and your Tca cycle. So what happens? So you have two molecules of pyruvate which are converted into acetyl-CoA. So you have two molecules of acetyl-CoA, right? Aerobic respiration in mitochondria, yes, you are absolutely right, okay? So your pyruvate is a 3-carbon compound, okay? And your acetyl-CoA is a 2-carbon compound. So here a carbon dioxide is removed. So this is a kind of decarboxylation reaction. So what is the enzyme that is getting used? Pyruvate dehydrogenase. So this enzyme, pyruvate dehydrogenase, is a multi-enzyme complex. So what do I mean by multi-enzyme complex? It has 4 coenzymes plus it has 1 cofactor. It is a huge unit. It is not a single subunit enzyme, but it has 4 coenzymes and 1 cofactor. So that 1 cofactor is Mg2+. So please remember, your pyruvate dehydrogenase is a very complex enzyme. It has 4 coenzymes and 1 cofactor. Okay. So the other 4 cofactors are lipoic acid, your NAD, thymine pyrophosphate, okay, and the last one is your coenzyme A, right? So these are the 4 core enzymes. So your enzyme pyruvate dehydrogenase is a multi-enzyme complex. It is made up of 4 coenzymes and 1 cofactor. So 4 coenzymes are your coenzyme A, lipoic acid, NAD, thymine pyrophosphate plus 1 cofactor, that is Mg2+, right? Yes, Munna, hello, I am very good. Thank you. How are you? So please do like, share and subscribe. Thank you so much for joining my session, Munna. Okay. Fine. So this is understood. Here in your link reaction, you have the production of 2 NADH molecules. So 2 molecules of pyruvate, so 2 molecules of NADH. Right? So this is the end point. So what is the net gain here? So here you have only 2 NADH. So converting this in terms of ATP, so it is 6 ATP. So in your link reaction, it is 6 ATP. In your glycolysis, it is your 8 ATP. Done? No doubt in link reaction? Yes. So Lakshmi Chandran, hi, yes, a very happy good evening. So thank you so much for joining my lecture. So please do like, share and subscribe. I am teaching you the process of glycolysis, link reaction, Krebs cycle. It is a revision because today I am supposed to start electron transport chain. Okay? Yes. So moving forward, so we are on with fermentation. So now I am talking about your Krebs cycle. So what happens in your Krebs cycle? So Krebs cycle is also known as Tca cycle or your citric acid cycle. Why? Because the first stable compound is your citric acid. So where does the process of glycolysis takes place? Sorry, where does the process of link reaction take place? So link reaction happens in your mitochondria. So because it is, why? Because it is a part of your aerobic respiration. So ma'am, 2 ATP only produced where? Where 2 ATP only produced? In case of link reaction, it is 6 ATP. Why only 2? 2 ATP is produced in case of glycolysis, you are right. So here 4 is produced, 2 is utilized. So 4 minus 2 is 2. That's why I wrote the net ATP gain is 2. Yes ma'am, a very happy good evening. So thank you so much for joining my lecture. So please do like, share and subscribe. Okay Antonia B, did you get? So 2 ATP is being used and 4 ATP is being produced. So 4 minus 2 is 2. So this is the net ATP gain. Okay? Yes? So moving forward, so then I spoke about link reaction. Now about your TCA. So TCA also takes place in your mitochondria. So all enzymes of, enzymes of your TCA, they are present in your mitochondrial matrix, that is the cytoplasm of the mitochondria, except one enzyme. So what is that one enzyme? That is your succinate dehydrogenase. So this is present in your inner mitochondrial matrix. Inner mitochondrial membrane. This alone has a different location. Okay, so moving forward, so all the intermediates in this process are used in your anabolic or catabolic reaction. So all the intermediates are being utilized. Fine. So I am not talking about the steps of your TCA, that is very complicated and not needed. So directly I am talking about the ATP gain. So we have one ATP molecule. So FADH. We have one, right? So this is your generation in case of your TCA cycle. So here the substrate is acetyl-CoA. So this is formed, this is converted into carbohydrate, carbon dioxide plus water. Yes, so this is the substrate, this is the final product, right? And your NADH, you have 2, right? And this is for one molecule of acetyl-CoA. So how many molecules we have? Yes? You're getting? So this is for one molecule of acetyl-CoA. So we have 2 molecules of acetyl-CoA. So it will be 2 ATP, 2 FADH and it will be 4 NADH. So this is 2 ATP, 2 FADH and 4 NADH at the end of your Krebs cycle. So converting everything into the form of your ATP. So 1 NADH is equal to 3 ATP, 1 FADH is equal to 2 ATP. So convert everything in the form of ATP. So it will be 2 plus 4 plus 12 ATP. So how many in total? How many in total? So 12 plus 4 is 16. So 16 plus 2 is 18 ATP. Yes? Okay, before I could read, you deleted it. Okay, Keshav? Fine. So this is understood? Yes, how we have 18 ATP molecules? No doubt? Yes. So for your total of your aerobic respiration, so we include your glycolysis. So glycolysis, it was 8 ATP. So your link reaction, how many we got in link reaction? So link reaction had 4 NADH. So it is totally how many? 6 ATP here. So moving further, your Krebs cycle or your Tca, it is your 18 ATP. So how many we have? So it is 26 plus 6. So how many we have? Yes? Quick. So you did we make a mistake? Did we make a mistake? Is it right? Yes. Okay. So 32 ATP we have in total. Right? Is this clear? 3 NADH. Okay. So from one molecule of, okay. Yes. Yes. I found out my mistake. So from one molecule of acetyl-CoA, we have 3 NADPH. Right? So 3. Yes, so totally for 2 molecules we have 6 NADH. So 6 into 2 is 12. So it is 6 into 3. So 6 into 3 is 18. So 18, 4 is 24. So 24 plus 2, it is totally 28, right? So am I right now? Yes, so it is totally 24 ATP. So plus 6. So 24 ATP in case of your Krebs cycle. That is what I was wondering, where I missed 2 ATP. So this is how you can easily get confused. So now what will be the answer? So we have totally 38 ATP. Yes, Antonia B, thank you. So at the end of your aerobic respiration, the net output is 38 ATP. Right? Now clear? So how we arrived at 38 ATP? So step by step it's very clear. Glycolysis, link reaction. So between link reaction you have fermentation and then you have your Tca cycle. Done? Yes? Your aerobic respiration is done? Okay. So now we move on to the next one. So this is your Tca cycle, your Krebs cycle. So here we have 3 NADH. So one here and one here, right? I assumed it as 2 and you have 1 FADH, okay? So please don't make this mistake. So the net equation is this. So here they have written 4 because they have included link reaction as well. Yes? Everyone is clear? So can we go to today's topic, that is your electron transport chain? Okay, so moving forward. So after your Krebs cycle, what happens? So at the end of Krebs cycle, yes, your carbon dioxide is formed, but energy also we need, right? So where is that energy coming from? So the last step after your Krebs cycle is your terminal oxidation. So terminal means the end, last oxidation. Now this oxidation is going to give us energy. Let's see what is this. So combustion of oxygen with electrons and H+ plus released from the reduced coenzyme produces water. So tell me, what is the end product of respiration? Yes, in respiration, I agree, you have the combustion of glucose molecule. So your glucose is completely broken down into carbon dioxide, water plus some amount of energy, right? So carbon dioxide is produced till your aerobic respiration in your Krebs cycle. So what about water? What about energy? So these two are produced by your terminal oxidation step, okay, the last one. So in terminal oxidation, so combustion of oxygen, so here your oxygen burns, okay, with electrons and your H plus ions released from reduced coenzyme, it produces water. So it includes two processes. So terminal oxidation is of two processes. One is your electron transport chain and the final one is oxidative phosphorylation. So let's talk about your ETS. So the respiratory process release and utilize the energy stored in NAD plus H plus and FADH2. So if you calculate, yes, okay Shimpi, yes, take care, okay. How it burns inside, it does not burn, it is broken down, okay. But technically we use the term combustion, right. So in respiratory process, so in glycolysis we had 2 NADH, right. In link reaction we had 2. In Krebs cycle we had 3. So for 2 molecules it is 6. So totally how much? So we had 10 NADH and 2 molecules of FADH produced, right, in your aerobic respiration. So energy is stored inside this. So how are you going to utilize this energy? Because we need energy in the form of high energy molecules. So we are going to utilize the energy stored in it. So accomplished when these are passed through your electron transport chain. So when your NADH and FADH passes through your electron transport chain system, it produces or it gives us the required energy. Electrons are passed to oxygen to form your water. So your electrons in your electron transport chain, your electron passes and the final acceptor of electron or the final owner of electron is your oxygen, which in turn produces your water. So now you understand where the oxygen that is produced by photolysis of water in your PS2 comes to. It comes to your electron transport chain in your mitochondria, because your oxygen is the final acceptor of electrons, which is ready to accept the electrons and in turn produce water. Okay, are you able to link it? Yes. So metabolic pathway in which your electron transport system is a metabolic pathway in which your electron passes from one carrier to the other, right? So it occurs in the inner mitochondrial membrane and it occurs only in eukaryotes. In prokaryotes, they do not have a membrane bound organelle called as mitochondria. So talking about the structure of mitochondria, it is a double membrane structure. So this is your outer membrane, this is your inner membrane and this is your inner membrane space. Inner mitochondrial membrane. This is your inner mitochondrial membrane and in the inner membrane space, okay, and this is your matrix. Fine. So the inner side of plasma membrane or the mesosomes in prokaryotes. In one news they said that there is a chance of new strain wave in 2026 and 2032. Okay, I would suggest let's all hopefully cross 2021 and then let's think about 2026 and 2032. Okay, yes. So how does this happen? So your ETS has 4 complex. So electron transport system is a network of few members. So here it has 4 members. The first one is complex 1, then you have complex 2, then complex 3, 4 and 5. So complex 1 is FMN NADH2 dehydrogenase. So FMN NADH dehydrogenase. Okay, yes. Because we are utilizing the energy in FADH2 in NADH2 and FADH2. Your NADH2 is conjugated with FMN. It is a coenzyme. Okay. Then you have your FADH2. It is with coenzyme Q. This is known as ubiquinone, right? And then you have your cytochrome complexes. This is B to C and this is A to A3, and finally you have your ATPase synthase. Just like in your chloroplast, in your chemiosmotic synthesis, you have your ATPase synthase, right? So similarly, the last complex is like this. So inhibitors, just give a glance, not very necessary. Meaning if I give amytol, this complex 1 will not work. Okay. So complex 1 NADH2 conjugates with FMN, complex 2 NADH2 conjugates with coenzyme Q, okay, and this is also known as succinate dehydrogenase complex, and number 3 is cytochrome B and C, 4 is A to A3, and finally you have ATPase synthase. So here we go. Who are the electron carriers? So in electron transport chain, electrons are carried, right? So who are the carriers? So that is your FMN. So that is why your NADH sends its electron to FMN. Then you have your iron sulfur clusters. Then you have your ubiquinone. So your NADH2 sends electrons to your ubiquinone. Then you have your cytochrome. Then your ubiquinone, cytochrome are major electron carriers. So cytochrome C is a small protein attached to the outer surface of the inner membrane. So hold on. So we'll look into the diagram and then talk about it. Yes, so this is your figure in your new version of your NCERT. Your old version of NCERT has a different figure. Now pay attention to this figure. So you have matrix, you have your inner membrane space, and this is your inner mitochondrial membrane. So it is like this. So I have my mitochondria. So this is my matrix. So that is written here. And this is my inner mitochondrial membrane, right? And this is the space. So the space, inner mitochondrial membrane, matrix. So where is your ETS present? Your ETS is present in the inner mitochondrial, in the inner membrane. Okay, so in the inner region of the membrane it is present. Inside you have a matrix, inner membrane space, and then you have your outer membrane. Okay, so you have your complex 1, complex 3, and then complex 2. So this is how it is arranged. So complex 3 is cytochrome. So the electrons from complex 1 and complex 2 flows into complex 3 because cytochromes accept electrons. Fine. So in case of your chemiosmotic hypothesis, so what we read, to produce energy there should be an H plus gradient, and when this H plus gradient is broken it produces ATP. So similarly here we are creating a H plus gradient. Here more amount of H plus gets accumulated in the inner membrane space. In case of your photosynthesis or chemiosmotic hypothesis, H plus ions get accumulated on the stroma side. Right, or sorry, on the lumen. Here it gets accumulated on the inner membrane space, and finally when this gradient breaks, so you have F0, F1. So similar to that in your chemiosmotic synthesis, so you have the production of ATP. So pay attention. So you have your NADH plus H plus is produced from your glycolysis link reaction, right, and your Krebs cycle. So when this is broken down into NAD plus, okay, when you have the reverse conversion, you have the release of 2 electrons, okay, and then it sends it to your FMN complex. They are carriers of these electrons. When your NADH plus H plus is converted into NAD plus, electron is released. So those electrons are given into the first complex which contains your FMN. Now when your FMN and FVS, that is ferredoxin ion clusters, combine, they release 2 more electrons. So here you have plus 2H plus, right? So here also it leads to the release of 2H plus. Okay, so when your NADH plus H plus from your glycolysis, Krebs cycle and ring reaction is reverse converted into NAD plus 2H plus. The H plus and the electrons released move inside. So the electrons are carried by FMN because they are electron carriers. So your FMN conjugates with FeS2. It once again produces 2H plus. So totally you have 2 plus 2, 4H plus. So that moves into the inner membrane space. But your electrons move via the electron transport carriers into your complex 3, that is your cytochrome complex. Now similarly from your FAD, your FADH2, so this is converted into FAD plus 2H plus. Right? So similarly this conjugates with your FeS and it produces 2 more H plus. So totally 4 H plus comes out here. So again H plus is getting accumulated and your electrons move inside the cytochrome. Okay? So from cytochrome BC it moves into A and A3. Right? So here you have the production of water. So water, so half O2 plus 2H produces H2O and here you have the release of 2 electrons. So you have more amount of H plus here compared to the other places. Thus a H plus or a proton gradient is produced. Now you have your F0 and F1 complex. So this is your transmembrane protein. Yes. So when your H plus moves across this, your ATP is produced. Okay. So 2 H plus are needed to produce 1 ATP molecule. So this is your electron transport chain. So did you understand? Should I repeat once again? Did all of you understand electron transport chain? Should I repeat it? Yes? Is it clear? Should I explain it once more? Antonia B. Keshav. Yes, Vairavan. Is this clear? How the process? Yes, okay. So Vairavan has understood. Others? Done? No issues? Antonia B? Is it clear? One more time. Okay. So in your electron transport chain, we are utilizing the energy that is present in your NADH and in your FADH. Okay. So here it is electron transport chain. So your electrons are moving inside. So as your electron moves inside, your H plus gets concentrated on the inner membrane space. So this is the matrix of mitochondria. This is the inner membrane. This is the space between the outer membrane and the inner membrane. So here your H plus ions get concentrated. So when your H plus ion concentration is present, that is a proton gradient, and then when this gradient is broken, you have your ATP production. Yes sir, a very happy good evening. So thank you so much for joining my lecture. So please do like, share and subscribe. Okay, right? Fine. So your NADH plus H plus is produced from your glycolysis, link reaction and your TCA. So this is reverse converted into NAD plus 2H plus. Right, and it also releases some amount of electrons. So the electrons are carried by your FMN, which in turn conjugates with your FeS. So when your FMN conjugates with your FeS, it once again produces 2H plus. So total you have 4H plus that is present. So this moves inside your intermembrane space. Hi Satyendra, yes, a very happy good evening. So thank you so much for joining my lecture. So please do like, share and subscribe. Right? So this is it. So now your electrons move into complex 3, that is your cytochrome. Now in complex 2, your FADH plus H plus is broken down into FAD plus 2H plus 2 electrons. Now once again when this FAD conjugates with FeS, it produces 2 more H plus. So totally you have 4 H plus which moves into the inner membrane space. Alright, yes. Now all the electrons move into cytochrome B, C and it comes to A. So in complex 4, you have the production of water, which is a product of respiration. So here half the oxygen plus half H plus gives you water, Yes, and 2H plus from here goes into your inner membrane space. Thus, you have more concentration on the inner membrane than on the matrix or the inner membrane. Okay, so thus a proton gradient is produced, and this gradient is broken down by your ATPase synthesis complex. So when 2H plus moves across this, so you have one ATP molecule formed. Is this clear? Yes. So half O2 plus 2H, it gives you water. Done? Is this clear? So 2H plus gives you 1 ATP molecule. So this is what is written in my theory. So I didn't read the theory before. So they are my electron carriers. So electron transport between the cytochromes 3 and 4. Right? So 3 is B and C, cytochrome B and C, and 4 is cytochrome A to A3. Right? So this is what I have been telling. So, role of oxygen is limited to terminal stage of the process. So, the presence of oxygen is vital because at the end it is accepting all the electrons and it is producing your water molecules, right? So, oxygen acts as your final H plus acceptor. So, cyanide inhibits the activity of cytochrome C. If your cytochrome C is inhibited, electrons will not flow inside. So, even if cyanide gets accumulated in plants sometimes, but your plant has an alternate mechanism. Even if cyanide is present, your ETS process continues. Okay, this is in case of your spinach and in case of your spisum. So, spisum is your P varieties. Okay. So, ETS I have explained. So, this is how it happens, NADH to NAD plus or H plus, mitochondrial matrix. So, this is your inner membrane and this is your inner membrane space. So, here you have more concentration of H plus and finally it is getting broken by your ATPase synthase complex. Done? So, everyone understood your ETS? Yes, for all those who said ETS is tough. So, I will talk about amphibolic pathway in the next lecture. So, don't worry, I am not saturating you. If only you have understood till your ETS from your glycolysis, that itself is a huge part. So, please don't forget to join me for my quiz at 6:30. It is guess the chapter. So, please prepare. So, it is a chapter that I have recently taught. So, that is the one clue that I can give you. Yes. So, I will meet you at 6:30. So, until then, stay safe and take care. And this is me signing off. If you genuinely liked my way of teaching, so please do like, share and subscribe. So, bye-bye. And this is me signing off. Bye.